Parkinson's disease. Is angiogenesis a possible treatment or even a cure? This presentation will address two questions. Number one, does Parkinson's disease result from a lack of blood perfusion to the dopamine-producing neurons in the brain? Number two, is angiogenesis, the natural regenerative process to grow new blood vessels, a treatment or even a possible cure for Parkinson's disease? Zhittya Regenerative Medicine is a biopharmaceutical company whose management over the last 20 years has expended in excess of $130 million advancing biological drugs which trigger human biological response known as angiogenesis and neurogenesis. Zhittya's management has advanced its drugs to trigger angiogenesis in the heart to treat coronary artery disease, in the feet to treat diabetic foot ulcers, and in the legs to treat peripheral artery disease. Zhittya's management has advanced the heart drug to U.S. FDA Phase 2 clinical trials. This presentation on Parkinson's disease, is angiogenesis a treatment or even a possible cure, is presented at a medical level for the general public to understand and is not meant to be an academic scientific presentation. The presenter is the President and Chief Science Officer of Zhittya Regenerative Medicine, Dr. Jack Jacobs. Dr. Jacobs.
Thank you, Dan. This presentation will be divided into 4 parts, and in the first part of this presentation, I want to review the evidence from a number of researchers that support our hypothesis that Parkinson's disease is initiated by vascular disorder in the brain. But before I do that, let's first look at what is currently known and generally accepted about Parkinson's disease. Parkinson's disease is a progressive disease, which gets worse over time. As this graph clearly shows, the devastating symptoms of this disease only worsen over time as this disease inexorably progresses. We know that in Parkinson's disease, a specific type of neuron is affected, and these are the dopamine-producing neurons. These dopamine neurons are located in the substantia nigra area of the brain, which is located deep in the middle of the brain. These neurons release dopamine, which is a critical neurotransmitter used in the brain for controlling movement. Presently, it is believed that when dopamine production by these neurons decreases by 80%, which is only 20% of normal, the symptoms of Parkinson's disease begin to be manifested. Now, this is a very important point to keep in mind during this presentation. Namely, if we can boost dopamine levels back up above this 20% of normal threshold, we have a chance to reverse this disease. Now, what are the primary symptoms of Parkinson's disease? As shown on this slide, they include stooped posture, tremors and shakiness, gait disturbances, rigidity and stiffness, and there's a general overall slowing down of movements of the body. What is not shown on the slide is that patients with Parkinson's disease also suffer from cognitive disturbances, including memory loss, trouble concentrating, and depression. In addition, they also have visual and smell disturbances. Please note that many of these symptoms are similar to what are experienced by stroke victims and those suffering from vascular dementia and Alzheimer's disease. What is the cause of Parkinson's disease? Unfortunately, the answer to that question is currently unknown. Now, certain risk factors have been identified for Parkinson's disease, as shown on this slide. These risk factors include certain inherited genes, concussions and brain injury, gender, more males get the disease than females, exposure to certain pesticides and heavy metals, age, and now let's look a bit closer at this risk factor of age. We know that the average age of a Parkinson's disease diagnosis is 60 years old. We also know that 5% to 10% of patients are diagnosed before the age of 50, and that these individuals are more likely to have an inherited form of Parkinson's disease. We know that once diagnosed at any age, a person will live with these gradually worsening symptoms for decades. At the bottom of the slide, look at the mean length of time these patients have to live with these devastating symptoms, from 21 to 38 years depending on the age of diagnosis. Although we know the symptoms, risk factors, and epidemiology associated with Parkinson's disease, we do not know why dopamine-producing neurons cease to function and cause this disease. However, there is a large body of medical research from neuroimaging studies and anatomical investigations which indicates that one possible cause of Parkinson's disease is vascular disorder. This disorder occurs in the substantia nigra region of the brain, which leads to a disturbance in the blood perfusion of dopamine-producing neurons and causing them to malfunction. This comes from peer-reviewed scientific and medical publications from a number of academic centers around the world, including such places as the University of Southern California, the University of Minnesota, the University of Rochester in New York, Cambridge University in England, and Tel Aviv University in Israel. Over 60 publications spanning a 20-year period support this hypothesis, and we would be happy to make these publications available to you upon request. As seen at the bottom of the slide, other neurodegenerative diseases, such as Alzheimer's disease, is thought to be initiated by vascular dysfunction in the brain. We believe that these publications support our hypothesis that vascular disorder leads to the demise of the dopamine-producing cell seen in Parkinson's disease. And as it will be discussed later in this presentation, we believe we can do something about it. Now, what is vascular disorder? Vascular disorder refers to a breakdown in the tiny blood vessels and capillaries that supply a particular tissue or organ, leading to a disruption of the blood flow. On the next slide is an excellent example of vascular disorder in the brain after a traumatic brain injury. This is what normal brain capillaries look like under a highly magnified microscope. And immediately after either a mild or severe brain injury, as seen here, these blood vessels are significantly depleted. Now on these two panels, you can see how the brain attempts to revascularize itself 14 days after a brain injury. Compare this vasculature to normal brain vasculature, and I think you can understand what the term vascular disorder refers to. It is this pathology which underlies the long-term disabilities that patients with a traumatic brain injury suffer. Now we know that vascular disorder and a lack of blood flow can lead to heart disease and heart attacks, peripheral artery disease in the legs, diabetic foot ulcers, and stroke and vascular dementia in the brain. Question: does vascular disorder affect dopamine-producing neurons in patients suffering from Parkinson's disease? Yes, we think it does. Imagine this type of vascular disorder in the region of the brain that contains the dopamine-producing neurons. Now, new and sophisticated brain imaging technology, such as functional MRI analyses, can measure blood perfusion in the brain. This is also referred to as cerebral blood flow, and here are some publications in that regard. Now, this technology can accurately and with great sensitivity measure areas of underperfusion in any area of the brain. And what have these imaging studies revealed? It has been shown that in normal subjects, there is a modest reduction in cerebral blood flow as we age, a 20% reduction by the age of 60 versus that of a healthy 21-year-old. In Parkinson's disease, it has been estimated that blood perfusion is decreased by up to 50% in the substantia nigra region, where the dopamine-producing neurons are located. In acute stroke, regional blood perfusion is decreased by greater than 80%. Common sense tells us that if any cell in the body is not getting enough blood, its oxygen and nourishment needs will be insufficient and the cell will suffer. So what do we know at this point? We know that with Parkinson's disease, patients can have up to a 50% decrease in regional blood flow. We also know that Parkinson's disease symptoms appear when dopamine levels fall by 80% or 20% of normal. Does this mean that 80% of the dopamine-producing neurons are dead and beyond rescue, as many medical experts believe? We think not. Let's look at stroke, where much more data is available to perhaps give us a hint on the fate of the dopamine-producing neurons in Parkinson's disease. Now let's look at stroke as depicted in this diagram. In the majority of patients, acute stroke is caused by a blood clot in one of the main arteries supplying the brain with blood, as shown here. It is well known that this interruption of blood supply leads to the death of neurons in the core region of the stroke, seen here, where the nerve cells die from a lack of oxygen, a lack of nourishment, and the buildup of toxic waste products. Now, an important point to make is that surrounding this core region is an area referred to as the penumbra, as shown here. This is an area where nerve cells become dysfunctional from the lack of sufficient blood flow. And remember, in acute stroke, blood flow to a region such as this would be decreased greater than 80%. So in here we would have underperforming neurons as well as hibernating neurons that are not doing much of anything. But the key point here is that these neurons are viable and they survive and lead to stroke recovery. We believe this is even more the case in Parkinson's disease, where blood perfusion is decreased by around 50% to the dopamine-producing cells versus the 80% in blood perfusion seen here in acute stroke. So let me summarize at this point. We believe that the fate of dopamine-producing neurons in Parkinson's disease is not binary, and that there are not only two choices to be had, either 100% functional or dead. We believe that similar to stroke, that dopamine-producing neurons in Parkinson's disease patients can also exist in an ischemic or hibernating state, and that they can possibly be rescued by restoring blood perfusion and vascular order to these neurons. We also believe that it is very possible that a patient with Parkinson's disease could have up to 40% of their dopamine-producing neurons functioning at a range of reduced levels, ranging from underperforming neurons to dormant neurons, and that these dysfunctional nerve cells are excellent targets for therapeutic intervention, as will be discussed later on in this presentation. Now it is time to introduce you to the concept of the neurovascular unit. As depicted in this slide, you can see that the brain is an incredibly vascularized organ. Now, if one delves more deeply into the smaller areas of the brain, it can be seen that these larger arteries branch off to smaller arterioles and then to capillaries. Each capillary supplies blood and oxygen to what is called the neurovascular unit, as shown here, that is comprised of a blood vessel, a neuron, and these supporting cells for the neurons. So critical is blood perfusion to the proper functioning of neurons that evolution has given each and every neuron its own blood delivery system. Billions of these units exist in the brain, and we believe a malfunctioning of the system is the primary defect in Parkinson's disease. Our hypothesis is that a lack of blood flow to the neurovascular units comprising the region of the brain where dopamine-producing nerves are located is the underlying cause of Parkinson's disease. We believe this is a rational and medically defensible explanation. We also believe that our drug that can trigger the regenerative processes of angiogenesis and neurogenesis could possibly treat this condition and perhaps even cure it. So now let's take a closer look at these two regenerative processes known as angiogenesis and neurogenesis. What is angiogenesis? As shown in this diagram from the Angiogenesis Foundation in Boston, angiogenesis is the growth of new blood vessels in damaged tissues. When a tissue is injured, it produces an angiogenic factor which stimulates a cascade of events as shown here. As I will discuss in a moment, the drug which we are developing for Parkinson's disease is an angiogenic growth factor. These factors bind to receptors on the surface of existing blood cells and through a series of biochemical and cellular events form these hollow tubes which mature into new blood vessels that bring nourishment and remove waste products from the damaged area. What is neurogenesis? Neurogenesis is the growth of new neurons in the damaged brain or other sites containing neuronal tissues. As seen in this slide, neurogenesis starts with neural stem cells, which then mature into neurons and other neuron-supporting cell types in the brain. Now, until recently, it was thought that adults could not regenerate their brain cells. But it is now known that even 80-year-olds have a reservoir of stem cells in their brains that can be used for regenerative purposes. It is also known that stem cells will avoid populating hypoxic or those areas of the brain that are underperfused with blood. It is almost like the neural stem cells can sense that survival in that type of hostile environment will be difficult. This is why the processes of angiogenesis and neurogenesis are tightly coupled to ensure that arriving stem cells are properly nourished and that their cellular waste products are efficiently removed. Thus, these two regenerative processes, angiogenesis and neurogenesis, can lead to the complete restoration of the neurovascular unit. To summarize this part of the presentation, our hypothesis is that a lack of blood flow to the vascular neuron units that comprise the region where dopamine-producing neurons are located is the underlying cause of Parkinson's disease. We also believe that our drug that can trigger the regenerative processes of angiogenesis and neurogenesis could possibly treat this condition and perhaps even cure it. To see if our hypothesis is correct, we will be advancing into U.S. FDA authorized clinical trials in 2018 and 2019 to see if a potent angiogenic and neurogenic human growth factor can reverse or abate the symptoms of Parkinson's disease. Now, in part 2 of this presentation, I want to discuss how a drug that stimulates angiogenesis and neurogenesis could be used to treat and possibly cure Parkinson's disease. When injury occurs, the body produces a very potent growth factor that can induce both angiogenesis and neurogenesis. This factor has been coined an angioneurin. The name of this growth factor protein is human fibroblast growth factor, or FGF1, and is depicted on the next slide. This is a depiction of the FGF1 protein. FGF1 is a simple protein in structure, much like insulin or human growth hormone. Over 30 years ago, I was a PhD graduate student in the laboratory at Washington University in St. Louis, where we first isolated the FGF1 molecule from the brains of cows. Since that time, I've been able to bioengineer the human form of FGF1, which is now produced in fermenters within a biomanufacturing facility, as shown here. This product has been approved by the U.S. FDA for use in human clinical studies. And if we go back to that slide on angiogenesis, it can be seen that FGF1 is that angiogenic growth factor that drives angiogenesis in our bodies. A key point here is that FGF1 initiates angiogenesis by binding to a cell surface receptor on endothelial cells, the cells that line all of the blood vessels in our body. Think of this interaction between FGF1 and its receptor as a jet, FGF1 being the jet, being hooked by a landing cable, the FGF1 receptor, on an aircraft carrier. And another important point to make here is that in ischemic or damaged tissues, the number of these cables are greatly upregulated. For example, in the ischemic heart, it has been shown that the FGF1 receptor is over 100-fold upregulated, and this allows the ischemic heart cells to be poised to react to any FGF1 that may enter this area. So think of ischemic tissue, whether it is in the heart or brain, as this drought-stricken field on the left here. Undernourished and starving, but with its FGF1 receptors tremendously upregulated. Then FGF1 is introduced into this field, much like dropping a match on the drought-stricken field as shown on the right here. Think of this as an explosion of metabolic activity leading to the formation of new blood vessels, and this wave of activity rolls over the ischemic region. And we will be dosing the Parkinson's disease patients every 3 days over a 5-month period. So we will be having this constant explosion of renewing metabolic activity as symbolized by the flames sweeping over the hills and dales of the ischemic tissue. Now, getting back to the heart, 4 human clinical trials have been conducted in which FGF1 was injected into ischemic heart muscle in patients suffering from severe coronary artery disease to trigger the growth of new blood vessels in the heart. I was personally involved in supervising 2 of those trials, which were FDA-authorized trials done here in the United States. Shown here is a paper published in 1998 from the medical journal Circulation, probably the world's leading scientific publication on the heart. This paper reported the first human angiogenesis trial in which German cardiovascular surgeons grew new blood vessels in the damaged heart with FGF1. Here is a picture from the article which shows the new blood vessel growth as seen during a heart angiogram. You can clearly see the blush of new blood vessels that appear 12 weeks after the injection of FGF1. Now, a very important point here is to keep in mind is that FGF1 only works in damaged or ischemic tissues. If we injected FGF1 into a healthy heart, we would see no new growth of blood vessels. This is because, as I mentioned earlier, that in an ischemic heart, the cell surface receptor for FGF1 is upregulated 100-fold. Since this initial work in Germany, over $130 million has been expended by us to advance the use of FGF1 in a number of medical indications characterized by poor blood perfusion, including severe coronary artery disease, chronic diabetic foot ulcers, and peripheral artery disease, which affects muscles in the lower leg. I now want to show you a short video that is a news clip from an ABC nightly news broadcast that highlighted the use of our drug in a U.S. clinical trial in heart disease patients. In this video, you will be able to see with your own eyes the growth of new blood vessels in the human heart after treatment with FGF1.
But since receiving an experimental treatment for his blocked arteries, his pain is gone.
I really feel great.
Duke was one of the first heart patients in the country to be treated with a protein actually capable of growing brand new arteries. The genetically engineered protein is injected directly into the heart. Within days, a network of new vessels begins to grow around the blockage, increasing the blood supply. Dr. Lynn Wagner showed us the changes in one patient's heart.
We see a small, narrow main artery and not very many secondary and tertiary arteries. This is after the treatment. What we're now seeing is new blood vessels growing here off the end of this artery.
And the patients themselves?
Symptomatically, they're improved within a couple of weeks of the treatment.
Just ask Constance Donnelly.
Oh, I feel wonderful. I've never felt so good in the last 5 years.
But doctors already see potential in other cases where the blood supply needs a boost, such as strokes and diabetes.
Now, in addition to our heart trials, over 10 years ago, we also initiated efforts to see if the FGF1 molecule could trigger angiogenesis in the brain as a treatment to enhance recovery from an acute stroke. More recently, we have initiated clinical development programs with diseases that we believe result from vascular disorder in the brain, including Parkinson's disease, Alzheimer's disease, and recovery from a chronic stroke. Now, as I mentioned earlier, about 10 years ago, we started our first effort in the brain with acute stroke. In this slide is animal data that shows that our FGF1 drug can prevent brain tissue loss after an acute stroke. You can see here that without FGF1 treatment, there is a large white area of infarcted brain tissue. However, with FGF1 treatment, you can see the significant reduction in this infarcted area that FGF1 affords in this animal model of stroke. The efficacy of FGF1 has also been examined in an animal model of traumatic brain injury. You can see here the injured area of the brain after a traumatic insult and how FGF1 protects and heals this lesion. So in both animal models of stroke and traumatic brain injury, it is very apparent that FGF1 is both angiogenic and neurogenic. I now want to show you some studies performed in animals that point to the promise of FGF1 being a potentially breakthrough new therapy to treat Parkinson's disease. Now, before the animal studies began, it was first established that FGF1 can promote the survival rate of dopamine-producing neurons in cell culture, as seen in this figure. Here you see a dose-dependent increase in survival of these dopamine neurons with the addition of FGF. At this dose of FGF1, you can see almost 100% of the neurons surviving. Animal studies have been performed in both rodent and primate models of Parkinson's disease. Here's animal data on FGF1 stimulating dopamine production in a mouse model of Parkinson's disease. In this model, the dopamine-producing cells in the brain are selectively damaged, leading to a drop in the level of dopamine in the brain and Parkinson-like symptoms in the mice. On the right-hand side of this figure, you can see normal levels of dopamine in the brain, and then the significant reduction in these levels when the dopamine-producing cells have been destroyed. So what happens when these mice are treated with FGF1? In the middle here, we can see almost 100% increase in dopamine levels in the brain over the untreated animals, approaching close to 70% of normal, as seen here. And here's some very important data in primates coming from a monkey model of Parkinson's disease. Now again, as with the mouse model, Parkinson-like symptoms were induced in the monkeys by exposure to this neurotoxic compound, MPTP. In this model, 9 separate behavioral abnormalities were scored, with a 0 score being normal and higher scores showing more severity of the symptoms. And these symptoms mimic those of Parkinson's disease, including such things as tremor and problems with gait, balance, and posture. On the left side here, you can see a dramatic improvement in these neurological scores after the monkeys were treated with FGF that becomes very apparent after week 10 of treatment. You can see a reduction in the severity of the symptoms by almost 1/2. This is very promising data, as good results in primates often predict success in humans. This next slide is a clear example of why I believe we are on the right track in looking at the use of FGF to treat Parkinson's disease. And before I explain what is on this slide, I need to talk a bit about the FGF family of growth factors. As it turns out, FGF1 is actually part of a large family that is composed of 22 closely related FGF proteins. I only had room to fit 9 on the slide. And there is not just one receptor for FGF. Think back to those landing cables on the aircraft carrier, but actually 7 cell surface receptors as depicted on this slide. This multiplicity of factors and receptors is what gives the FGF family so many diverse biological activities. Now, FGF1 is considered the mother of the FGF family, as it can interact fully with all 7 of these FGF receptors. Look across this top row, where it can be seen that FGF1 and only FGF1 can interact with 100% activity with all 7 of these FGF receptors. No other member of the FGF family can do this, and here you see only 1% activity of these FGFs with this particular FGF receptor. So it is this full activity across this whole range of receptors that gives FGF1 its potent and diverse activities, including stimulating both angiogenesis and neurogenesis. Now, if we return to this figure in the brain, we see that this cousin of FGF1, FGF20, has a remarkable and specific location in the body. FGF20 is primarily localized to the brain compared to other organs in the body. And in the brain, it is remarkably localized to the substantia nigra compartment of the brain, where it has been shown to promote survival of the dopamine-producing neurons. Now, this cousin of FGF1 has been shown to interact with only one of the 7 FGF receptors, so FGF20 appears to be mainly a neurogenic factor without a significant effect on angiogenesis. Our FGF1 molecule fully activates the same FGF receptor as FGF20 does, which is located on these dopamine-producing cells. But unlike FGF20, FGF1 can also potently drive angiogenesis. So again, I would like to emphasize that we believe FGF1, being the mother of all FGFs, will do a better job in treating Parkinson's disease. But I think the fact that this very tiny area of the brain that contains these dopamine neurons has its very own resident FGF20 molecule points to the importance of the FGF family in maintaining the viability of these dopamine-producing cells. Now we have prepared a white paper on the role and potential use of FGF1 to treat Parkinson's disease, and we are happy to provide you with this paper. In addition, as I mentioned earlier, we also have the original papers to support all of the statements and studies that have been referred to in this presentation, and these are also available upon request. Just briefly, I also want to touch on the fact that another neurodegenerative disease, Alzheimer's disease, is also thought by many experts to be initiated by a lack of blood perfusion to specific areas of the brain. In probably the largest study of its kind, medical researchers at McGill University in Montreal, Canada, followed over 1,100 patients with Alzheimer's disease and plotted the development of abnormalities with biomarkers and cognitive testing. What this remarkable study showed, as seen in this figure, is that the very first symptom which appears in these patients is a blood perfusion defect, as shown by this brown line. This decrease in blood perfusion precedes all other symptoms of this disease, including the appearance of amyloid plaques in the brain, this line here, and memory loss, shown by this green line. This is why we have expanded our clinical development efforts to explore if our FGF1 molecule can possibly be a treatment not only for Parkinson's disease, but also for Alzheimer's disease. Both of these diseases have few, if any, meaningful therapeutic options, and both are classified by the FDA as unmet medical needs. Another point I would like to make here is why the brain does not naturally regenerate itself in Alzheimer's disease and Parkinson's disease. Researchers are not exactly sure why, but they believe it has something to do with the nature of the trauma at a cellular level. Both Parkinson's disease and Alzheimer's disease follow a slow and insidious progression. There is no acute traumatic event, like with cutting yourself or breaking a bone, which would signal the body to produce FGF1 to regenerate itself. We believe there's a gradual and incremental vascular disruption occurring slowly over many years in Alzheimer's disease and Parkinson's disease, and we believe this is what results in the brain's neurons becoming dysfunctional. We also believe that this slowly evolving vascular disorder in the brain does not create a traumatic injury with a high enough threshold to signal the brain to make FGF1 to heal itself with angiogenesis and neurogenesis. Now, in part 3 of this presentation, I want to go over our plans to develop FGF1 as a potential therapy for Parkinson's disease by undertaking FDA-authorized clinical trials. Step 1 of this process is that we are currently carrying out toxicity studies in animals to be sure that long-term subcutaneous injection of FGF1 is safe and well tolerated. We plan to expose human subjects to FGF1 for up to a 5-month period, and we will be doing the same in these animal studies to look for any toxicities or side effects of our drug. We are very fortunate that a number of studies have shown that FGF1 can cross the blood-brain barrier after subcutaneous administration, so we will be able to use insulin pumps to slowly administer our drug to Parkinson's disease patients over an extended period of time. Step 2 of this process is that after completion of this animal study, we plan to initiate U.S. FDA-authorized trials at different locations in the United States in 2018. The Phase 1 clinical trials will be utilized to establish a safe dosing regimen. We will have an idea of this from our animal testing, but experience has shown us that in humans it can be different. We will be very cautious and start at a relatively low dose level of FGF1, administered over short periods of time. We anticipate doing many of these Phase 1 studies with a small cohort of patients, say to 10 to 15 per dosing regimen, until we find a safe and well-tolerated dosing schedule. We will also be monitoring efficacy in these trials, which is the improvement of symptoms, but safety, not efficacy, will be the primary readout from the phase 1 trials. It is in the phase 2 trials that efficacy will be the primary readout, and we also anticipate doing multiple phase 2 clinical trials to explore what works best for Parkinson's disease. If we see any hints of efficacy with our therapy in either the phase 1 or phase 2 trials, we will immediately apply for breakthrough therapy designation from the FDA, which will allow an expedited approval process for FGF1, including possible FDA approval after a phase 2 clinical trial. The traditional drug development pathway involves a subsequent phase 3 trial in an even larger patient population, which can add 2 to 5 years onto a clinical development program. In summary, do we believe that FGF1, by stimulating the processes of angiogenesis and neurogenesis, can possibly slow or even reverse the symptoms of Parkinson's disease? We are very hopeful that it will, and we are committed to bringing this potential therapy into clinical trials in 2018. We have some strongly held beliefs that are backed up by scientific and medical evidence that underlie our hope that FGF1 will be a breakthrough therapy for Parkinson's disease. These include, like with other areas of ischemia in the body, we believe that in Parkinson's disease, the dopamine-producing neurons in the substantia nigra become ischemic and are being starved from a lack of blood perfusion. That these neurons have highly upregulated their FGF receptors in a cry for help, and that therapeutically administered doses of FGF1, when it arrives to those starving neurons, could generate a very robust angiogenic and neurogenic response, maybe at even a low dose. Remember, in the ABC News clip, the woman's heart that was shown to have grown new blood vessels only received 1/10 the optimal dose of FGF1 that we established in prior animal trials. And also remember that she had an improvement in her symptoms within 6 to 12 weeks. It would be very heartening to see a similar response with Parkinson's disease. We also believe that the fate of the dopamine-producing neurons in Parkinson's disease is not binary, 100% healthy or dead, but includes a significant percentage of ischemic and hibernating neurons, similar to what was seen in stroke. We believe that by reperfusing these neurons with blood, that we can drive dopamine levels up past the 20% of normal threshold to reverse the symptoms of Parkinson's disease. We also believe that therapeutically administered doses of FGF1 will also be neuroprotective and recruit neural stem cells to repair the site of injury in the substantia nigra. Now, in the last part of this presentation, Part 4, I want to tell you how you can be kept informed of our progress and also how you can personally receive additional information from us, such as our white paper on Parkinson's disease or other publications. We will be starting a blog for Parkinson's disease, and you can visit that blog to get up-to-date information on our activities, as well as with the activities of other Parkinson's research groups that may have competing ideas to our own. In addition, the blog will keep a close watch on ongoing clinical trials being done in Parkinson's disease. We also have a form you can fill out if you would like to be considered to participate in our clinical trials. Finally, if you have an issue you would like to discuss with us directly, do not hesitate to call us at the number on the following slide. Here is our contact information. I want to thank you for taking the time to listen to this presentation, and please do not hesitate to contact us. Thank you again.